I'm happy to sit there. Well, thank you all. I'm Rick Carlson. I'm the director of the Department of Terrestrial Magnetism, which is one of the two Carnegie departments on this campus. This is the kickoff event of our spring neighborhood lecture series. We have a break during the winter, and the reason for that is we don't want to subject you to coming out here in cold weather with ice and snow all around. We'd rather invite you out here when it's spring and you can see all the blooming flowers and the beauty of our campus. So that was the thought, and hopefully by the next time you will be able to enjoy that. This is it. You know, what can you do with weather? So we have what we have. It actually turned out to be not such a bad day. So for those of you who've been to the neighborhood lectures before, you know that there are two Carnegie departments on this campus. Mine, the Department of Terrestrial Magnetism, and also the Geophysical Laboratory. We do work in sciences ranging from material sciences to earth and planetary sciences here. But what you probably don't know is that Carnegie supports 4 other departments scattered around the U.S. One of them works in the field of embryology. It's on the campus of Johns Hopkins University in Baltimore. We have 2 departments on the campus of Stanford in California. One investigates plant biology and one does global ecology. And then we have the Carnegie Observatories, whose headquarters are in Pasadena, and they run and manage a large telescope facility in the Southern Hemisphere in Las Campanas, Chile. So this group of 6 departments of the Carnegie is managed locally by directors, but overseen by the staff down at the headquarters of Carnegie down on 16th and P Streets downtown. It's managed by a board of directors, but also, most importantly, the president of the institution. It's my great pleasure and honor today to welcome the current president of the Carnegie Institution, Matt Scott, who will be our lecturer tonight. So Matt became the 10th president of the Carnegie Institution in September of 2014. So he's been with us now going on 2.5 years. He came to us from Stanford, where he was a professor in the, let me get this right, you got a long department name there, Matt. It's the Department of Developmental Biology, Genetics, Bioengineering, and Biology in the Stanford University School of Medicine. Matt did his undergraduate and graduate degrees both at MIT. After a postdoc at the Indiana University, went on to a professorship at University of Colorado, and then moved to Stanford in 1990. At Stanford, he was the chair of the biology department for 6 years, and he was chair of the BioX program, which is an interesting multidisciplinary effort to combine life sciences, engineering, physics to understand the workings of the human body. He was chair of that from 2001 to 2007. Matt's a member of the American Academy of Arts and Sciences, the National Academy of Sciences, and the National Academies of Medicine. He won the Young Scientist Award from the Bassano Foundation in 1990, and this recognition of a young, distinguished, and looking good scientist was recognized in 2013 by the Pastoral Cancer Research Award. So that word should tell you something about this. If you've been to previous lectures here, you know that on this campus we study things like, I can tell you how old the oldest rock on Earth is, we find exoplanets, we do interesting things like this, but this man works on cancer. He works on the cure for cancer, and not only works on it, he's actually had some progress on it. It sort of puts our work in physical sciences into a different context. So Matt's work focuses on the genes that control the embryonic and later development that's governed by the proteins that regulate the communication between cells and their growth. His research addresses how damage to the genes leads to birth defects in cancer, and as I mentioned, he's working on cancer cures, and one of them is that he found the genetic links to the most common form of human cancer, basal cell carcinoma, which those of us who used ourselves when we were young by spending too much time in the sun know too well. And then he also has investigated the most common childhood malignant brain tumor, medulloblastoma. So the subject of his talk tonight begins with a study from another Carnegie scientist, Barbara Mcclintock, who in 1983 won the Nobel Prize for what was called jumping genes. It was a discovery through her work of how chromosomes change during the reproduction of corn. So tonight, Matt is going to take us from that initial start and explain how these jumping genes play a role in everything from the development of life on Earth to the concerns about human health in his talk entitled Jumping Genes.
Very nice, quite excessive. All right, so we've got to make them jump. So could we have the lights down a little bit?
Turn the music off.
The music's going off, yep. Don't worry. It is, there it goes. Okay, so we're telling a story today about how studying some very particular thing, this happens often in science, you look hard at something peculiar, something special, something you initially think probably only applies to the organism you're looking at, and you discover something with immense generality that changes completely the view of how genes work and how evolution works and disease processes and so on. And that's what happened with these jumping genes. Starting, as you heard from Rick, with Barbara Mcclintock. Actually, it started prior to Barbara Mcclintock, but she's the one who really figured out what was going on. And she was working on corn, primarily, not only, and looking at chromosomes. And many of you have seen stained chromosomes. That's a cell of a lily splitting into two cells with the deep purple chromosomes stained and sitting on this skeleton of fibers. And they're separating, perfect copies, separating into the two daughter cells. Here's more, and I want to just point out that these corn kernels have little sectors, little fine stripes, little patterns. And Barbara Mcclintock was studying those little fine patterns and noticing some very strange things going on. So I want to lay out what I'll tell you about. But before I forget to say it, I mean, I'm really grateful that you came to join us tonight. I'm really flattered to see so many people here. This is a tough topic, but I've been told you're really smart. So what I did was to put the worst stuff at the beginning so that we can get through it and you can forget about it or whatever, or at least get a good nap later. So here's what we're going to do today. We're going to have a little introduction to the genes and proteins, to what they are and how they work. Very superficial, really, but hopefully enough to power you through the rest. That's the really tough part, so it's in red, because it's like a warning, okay? Then we're going to talk about genetic maps, how you look at where genes are along a chromosome, and look at the fact that most of the time they stay put, as they should. But then there are genes that jump, called transposons, because they transpose to a new place. That's the topic. Those are the jumping genes. And then we're going to look at a few different consequences of those jumps, those patterns in corn you just saw, so that's plant variegation. A lot of your house plants are very good at this. Consequences in terms of antibiotic resistance, transmission through bacteria, consequences for humans. Then we're going to look at the relationship, which there is, between transposons and viruses. So that's strange. What do they have to do with each other? And then we'll talk a little bit about evolution and how over the billions of years there have been some forms of life on Earth. These genes were, these jumping genes, these mobile genes, were clearly responsible for a lot of rearranging of the furniture, as it were, and changing things in the genes that would lead to changes in the form and function of microbes and of plants and animals and fungi and so on. Now, your genes are contained, as you know, in each of you has 46 chromosomes in each cell. They come in pairs with one exception. This is a woman because there are two X's, so there's no Y here. And you get one from dad, one from mom, and you are a kind of blend, as you know, of the two. When you spread them out from a single cell, they look like a mess, but using stains and then cutting it, literally cutting with scissors or with Photoshop, you can line them up in their pairs and recognize what's there, and you can see changes in them. You can see damage to them. You can see, there aren't any cases of that here, but you could see a case where one of them is broken off and a piece of it is stuck onto another one. That kind of mutation, that kind of genetic change is not all that rare, and it's stimulated by things like X-rays, for example. And Barbara Mcclintock was studying in corn some of those kinds of peculiar chromosomes that were breaking and pieces were sticking to other pieces and so on. And it was the beginnings of her discovery. She made many discoveries, but she probably deserved a Nobel Prize before she discovered transposons, but this was her sort of crowning glory. Now, if you take one of these stubby little things, they're actually immensely long strands of DNA. So if you spread them out, like this one, there's a chromosome sort of falling apart in detergent, and maybe those of you in front anyway can see that there are all these fine strands. It's an incredible tangle. In fact, in each one of your cells, and each of your cells is nearly invisible to the naked eye, if you stretched out the DNA from your 46 chromosomes, it would be about as long as from my fingertips to my fingertips, about 2 meters of DNA in each of your cells. So, and folded up, you know, it looks like the familiar chromosome, but stretched out, it would be that long. So you can imagine that you have to copy all this stuff and separate it, as I was discussing with that picture of a lily cell dividing, into 2 cells. So you've got to somehow deal with the tangles. And if you think you've had a bad hair day, just imagine 2 meters of DNA packed into a tiny space, and you're going to copy all of it and then spread it into 2 daughter cells. It's an amazing process. And in 1953, the chemistry of that process was figured out in the famous work of Watson and Crick, who determined the structure of these fibers of this DNA that makes up these chromosomes. And here's a diagram of what they deduced. Okay? It's the famous double helix, and it's made up of 4 little chemicals strung together in a long string. That's the string we've been talking about. The 4 chemicals are abbreviated A, G, T, and C, and they're color-coded here. And one is on each strand, and they pair up. And an A is always opposite a T, and a G is always opposite a C, as you can see in this diagram. Now, if you took this double helix and like a zipper ripped it apart, and then started using this relationship to rebuild the double helix by adding things on. Wherever you found a T, you'd stick an A on it. Wherever you found a G, you'd stick a C on it. And you could do that here, and you could do it there, and you see how you end up with 2 perfect copies of the original, which will then separate into the 2 cells. That's how you copy your genes. Okay? I warned you this was going to be the hard part at the beginning. Okay? It's going to get worse before it gets better. I should warn you. Okay? So this sequence of A's, C's, T's, and G's goes on for billions, okay, down the chromosomes. Each of your 46 chromosomes has about 1,000 genes in it. A gene is a section of this code. Okay? And genes come in different sizes. They're not very regular. Okay? Any questions so far? You should interrupt with questions, because otherwise I might keep talking. Yes?
How do you pull them apart?
There's a machine. The question is, how do you pull them apart? There is a machine. This doesn't happen spontaneously. There's a machine made of proteins that separates them and does the copy. The technical name for it is polymerase, DNA polymerase. It makes a polymer.
It's a natural thing that happens. It's not a human being.
It's a natural thing that happens, and you're doing it right there. For instance, with your blood cells, you're making new blood cells right now. And with your skin cells, you're making new skin cells. Your hair is growing. Every time you make more cells, this has to happen. Every time a cell divides into 2 cells, the DNA gets copied. Nearly perfectly, yes.
But to follow up on her question, it can be induced.
It can be induced, yes, absolutely. For example, if you cut yourself, the skin cells along the cut will start to divide. And other skin cells will divide differently away from the cut.
Or we can just directly add polymerase.
You can actually do this in a test tube by adding polymerase. Yes, you can make an extract and you can get this to go. That got a Nobel Prize for Arthur Kornberg of Stanford. Yes?
I've seen this illustration or things like it hundreds of times.
The blue ribbon is a chemical chain of sugar and phosphate alternating. Simplified here just to, yeah, that's the sides of the ladder, and in a way the ACs, Ts, and Gs are the rungs of the ladder. Then twisted, yes?
You use the number of 1,000 genes. Is that a new up-to-date number, or because normally you see a much larger number than that?
1,000 genes per chromosome. There are 23 chromosomes. So we have estimated, depends what you call a gene, but about 20,000 genes. The history of that is that when some of the simpler organisms, what we thought of as simpler organisms, were first analyzed for their genomes and genes were counted, they had 10, 20,000 genes. So there was this sort of assumption that because we're so complicated, we'd have more genes, which is the same sort of logic as saying the best bicycle for you is the largest bicycle. And in fact, when you tracked the publications for the number of genes in the human genome, it started at 100,000. And over time, as people did better analyses, it kept coming down. In fact, if you extrapolate, it would go to 0, but that's not what happened. So we have about 20,000. Yep. Okay. Now, this is a polymer, meaning it's a chain of sort of like units repeating in various patterns. The sequence of these things is different in different genes, and that sequence is the genetic code. That code is why you look like your parents. That code is why you look like a human, not like a turtle. That code is the instructions for building a brain, the instructions for building your immune system and your liver and your kidney and everything else. And there's a lot of work going on, including in our departments that do biology, about how that code works. Because if somebody tells you they really understand how the shape of your vertebrae is encoded in this code, they're lying, okay? It's not true. We know a lot about it, but we certainly don't know the full story. It's a very active area of research. Okay, but what we do know is that one of the things this code does is provide the instructions for how to build proteins. You have tens of thousands of different proteins in your body. And you notice tens of thousands isn't all that different from 20,000 genes. And indeed, the simplest way to say it is that each gene carries the code, the information, for one kind of protein. Now, proteins are gorgeous. This, while it's okay, is not, the hot date. But look at these things. I mean, these are beautiful, okay? Now, this is false color, of course, showing the molecular structures of a little sampler of some different proteins. And you can see they have much more elaborate structures, three-dimensional structures. These are machines of your body. And there are thousands of different ones, of different shapes and sizes. And they do all sorts of different work. In the analogy of building a house, some of them are bricks and mortar, some of them are framing, some of them are carpenters, some of them are architects. Proteins do all sorts of different functions in your cells, okay? And since you have to build a lot of these things, you need instructions, and the instructions are in the DNA. So let's just say what I just said. They can be motors, actually, like the movement of your muscles is a protein. It's actually a set of proteins burning basically burning sugar to move. They're motors. And in one that's familiar to you, this one, which is called hemoglobin, it's a protein that looks red to us often. It carries oxygen. It binds oxygen. It's in our red blood cells. It's what makes them red when there's oxygen on them. And it carries oxygen from a place where there's lots of oxygen, the lungs, moves in, carried in its blood cells through the body to a place where there's low oxygen. The protein changes a little bit its shape in a low oxygen place, and the oxygen falls off. It's delivered its stuff, okay? That's just an example of one protein. Now, all these proteins are themselves polymers. If you pull it apart, they are a long, linear polymer, like DNA is, but made up of different little beads on the string. And these are the beads. In DNA, there are 4 different flavors. Here, there are 20 different flavors, and their chemical names are listed down here, and I expect you to memorize them. Okay? There are 20 of them, and they're called amino acids, and you eat them. Your body is able to make some of them, and some of them you have to get through your food. And when you digest, you eat proteins from various sources. Your body breaks up the proteins into amino acids, and then you can use them to build your own proteins. Should bet. Okay, so you see by the colors that these chains, these proteins can be long, they can be thousands long, and at each position you can have any of the 20 amino acids. And since the 20 amino acids have different properties, the resulting chain will have different properties and will fold up into some shape that does something useful. If you make a protein that has some random sequence of these amino acids, it won't do anything. It won't work, it won't. Okay, so over billions of years of evolution, there's been a code selected to build proteins that matter for something. Probably starting with small ones and then eventually larger ones. Okay? So now the question is, how do you control the building of a protein inside a cell so that you put things in the right order? Because that's really all you need to do to make all these machines and building blocks and so forth. Okay, so we're at the hardest part here. How are we doing? Anybody still with me? Both of you are. That's really great. Okay. So there's a code, okay? And you've got a line of DNA things, A, C's, T's, and G's. You've got a line of amino acid, and that's these 20 different things. So it can't be one-to-one. A can't represent one of these amino acids because you'd run out, right? You'd quickly run out. You only have 4 options for DNA. 2 of them. What about 2 of them? Well, they're 4 in either position, right? So 4 times 4, that'd be 16 possibilities, but you've got 20 of these. You're still in trouble. So the code can't be 2 things. How about 3? 3 works. 3 gives you 4 times 4 times 4, 64 possibilities. And here is your genetic code. First, look over here. The letter, I've substituted U for T here because there's a processing step where you get a different, slightly different chemical, but it's a lot like a T. And if the first letter in a triplet of the DNA is A, and the second letter is U, so that's these 4 things, and the third letter is, let's say, C, you got AUC, and you put in that particular amino acid in line. So that's the code that was worked out in the 1960s that is the basis of all life as we know it. It's an amazing thing because it's kind of elegant and simple, okay? But let's look at it a little more. At the top, I've put a little piece of, you can think of it as DNA, okay? And there's a line of those subunits, those like beads on a string of the DNA. That's one side of a double helix. And now we're going to build a protein sequence to go with it. And the first thing there is UUC. So we look over here, and UUC is up there, and it says Phe, Phe, which is one of the amino acids. So we're going to plug that in. Okay, the next one is GGG, and that's glycine. And the third is UGC, and that's cysteine, and so forth. You see how the code in the DNA guides the assembly of the chain of amino acids for the proteins. Whew. This is our real low point here. Okay.
So what happened to the other side of the helix?
Yeah, the other side is pulled apart, and a copy is made of just one side, and for this process, you just use one of the sides. Okay? And there is a machine, as you were probably about to ask me in your follow-up question, there's a machine that does this. It's a very complicated machine called the ribosome. It has lots of proteins in it. It's a huge machine. Yes?
What allowed the person in the 1960s to be able to do this that they hadn't been able to before?
Well, one key thing is that biochemists could make extracts, and it would happen in the extract. So you could feed it different gene sequences in the extract and see what protein was made. So you can synthesize test sequences. For instance, something that was all G's would be gly, 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 gly. Okay, aha, that's the code. And then you try another one, and so on, and figure out what you got until you know all 64, what they do. Okay, and you can see there's some redundancy, like all 4 of these triplets all mean the same thing. And then there are a couple that mean stop. There are 3 that mean stop. Enough already. Right? What you're feeling right now. Okay. All right. So that's it. This is the worst. This is, this is as hard as it's going to get, I hope. Because what I've shown you is that chromosomes are made of DNA. DNA is a long polymer that carries a code, and that code is interpreted, each gene coding for a line of amino acids that makes a particular protein that does a particular function. All right. So, just to give you time to let that sink in a little bit, here it is in writing, so it must be true. Okay. A chromosome is a long piece of double-stranded DNA, a ladder twisted into a helix. Each DNA strand is a chain of A, C, T's, and G's. A on one strand is matched with T on the other, and C with G. That's how you copy it. A machine, which I didn't mention, called RNA Polymerase, makes a copy. This is an answer to your question, really makes a copy of one of the strands that has the U's instead of the T's. That's RNA. And then this other machine I mentioned, this huge one called the ribosome, reads the genetic code in that single-stranded thing to assemble amino acids into a protein. And so here's another example of that, translating the genetic code in triplets. Okay. Now let me just point out, yes, you had a question.
I just wanted to say, maybe saying how this RNA breaks down and starts to divide would be a great idea for people to know how important magnesium is in that process.
It is, it's very important. It's also magnesium and other small ions like that are very important for the structures of proteins as well. So these things, all sorts of, these molecules breathe and flex and change shape, and things like salt concentrations and temperature really change their shape. So if you boiled eggs, you know that that protein turns from clear to white, and that is an unfolding, a denaturation it's called, of the protein in the egg. The egg white is getting white. Okay? So I'll just mention that what mutations are, we hear that mutations are caused sometimes by sunlight, by ultraviolet radiation, by X-rays, by certain chemicals. They are changes in the DNA sequence of every kind you can imagine. Substituting one thing for another, sticking some extra stuff in, taking some stuff out, all of those kinds of things happen. And as a result, the sequence of amino acids you'll assemble will go crazy. Even just sticking one thing in here means all the others will be shifted, and they will be gibberish because you'll be reading the wrong sets of 3. Okay? That's what mutations can be. Now let's go back to looking at a chromosome. Remember, a section of a chromosome, a part of the DNA helix, is a gene. And in one chromosome you might have 1,000 of them, coding for about 1,000 proteins. All right? So that's our starting point, and as I've said already, since you have about 20,000 genes, you can make about 20,000 proteins. It's actually much larger than that because there are ways that you get variables stuck in. And proteins are a big part of who you are. About 17% of your body weight is protein, depending on Haagen-Dazs consumption and a few other things. Okay, so let's go back to here. And I know it's going to seem awful that all we've done is get through the first one. But actually, we're okay because the others aren't nearly as difficult as the first one. Okay, so we're going to talk next about genetic maps. Now, what I've told you is that a chromosome, although it's all tangled up into this lump, actually, if pulled open and stretched out, would be a single line with ends. And by the way, Barbara Mcclintock did the most important early work in understanding that those ends are very special. They're sort of caps. They're called telomeres. Okay, so she contributed in many different ways, and she keeps coming back. So we're going to talk about genes that don't jump, because as is often true in science, you start by establishing some principles that make a lot of sense and seem to be upheld by all the evidence, and then along comes somebody who says, wait a minute, it's not what you thought. It's not as stable as you thought. But we have to start with something that's stable, which is historically what happened too. And something that's quite stable is a map of the Washington Mall. If you're a tour guide, you can reasonably expect things to stay where they are. We're going to draw a line and we're going to make a linear map of a few items on the mall. Okay, those three. All right, and that's their relative relation. And it would be deeply disturbing to a tour guide to come and find that this order was no longer true. Okay, so we're going to color them in, and we're going to put that up there and get rid of the original map and just talk about maps like this as an analogy for genetic maps, which locate genes. Okay, so we're going to have a chromosome here, and we're going to say we're representing a section of it, which is more than one gene. So the magenta here shows a gene, but this keeps going. And so we've got room here for other genes, and we might as well give them the same colors. Okay, so we've got genes A, B, and C, not very clever, but you've got to start somewhere. Okay, so that is a genetic map. It just shows where along the DNA the genes are sitting. And we're not going to worry about what these genes do. This one might be to make eyes, dark, and this one might be to make a bone a certain shape, and another one might be to give you good musical ability or something, right? They have all sorts of functions. Usually they act in combination. Okay, so if you take this situation and you have established, and I'm sparing you how you make these maps. You can talk about that after the talk if you haven't had enough suffering. But there are elegant ways of making the maps. And so you know where the genes are. And if you look in one person, you'll find the genes like that. And you look at another person, and you will find the genes like that. And you look at another person, and you will find the genes like that. And you look in a chimpanzee, and it might be a bit different. Or it might be the same, because we're very close to chimpanzees. If you look at a turtle, they're more likely to differ, and so forth. The farther away you get, the more things are likely to change. Why should that be? Why wasn't there an early animal, put all the genes in the right places, and just keep them there? And there are two facts. One is the genes will still work if you move them around. And the second fact is that things do move around. And that's how evolution happens, and we'll come back to that. So in this situation, there's a nice normalcy. But in this situation, you look at person 1 and 2, and they're what you expect. And here comes trouble. And you look at person number 3, and they have a change, which if you think that's normal, and there's always an issue of what you define as normal, then this is a mutation where a gene is out of place. Okay? So that is disturbing because something happened. What happened? And this is in essence what Barbara Mcclintock started to find in certain circumstances. Here's the Washington Monument, and here's the Capitol, but there's Lincoln Memorial going for a walk. Okay, and that's really upsetting if you're a geneticist too, not just if you're a tour guide in DC. Okay? It gets worse because in certain situations, you get a situation like this where a gene has disappeared from its normal location. You see it over there? And it stuck itself into the middle of another gene. That is really upsetting. It's sort of doubly upsetting. So it's as though the Washington Monument, which is supposed to be there, has done that. Okay? And what are the consequences of that kind of rearrangement of the DNA? So one consequence is that you're missing gene B. It's over there now. So you're not missing it altogether, you're missing it from its normal location. It's inserted into gene C. Okay, and this is a bad thing. And gene C no longer works because in the middle of what should be the protein coding stretch of ACs, Ts, and Gs, you've got this bizarre sequence that landed from outer space, right? Over here, you no longer have gene B, and so it's certainly not working here, but it might be working just fine embedded in there. It might still work fine. Depends a little bit whether it was precisely chopped and so on. So a gene with the ability to jump like that is called, as I mentioned earlier, a transposon because it can transpose to a new place. And there are variations on this where it copies itself. So it's still here, but it's also here. You have a duplication of the gene, which sometimes doesn't matter at all and sometimes does something. Okay, we're going to take a breather and summarize some of what you just heard and where we're going. So first of all, most genes stay put most of the time in specific locations in any particular organism, in any particular species. Some genes do not stay put and at high frequency can jump to new locations, even during the growth of a plant or animal. The genes that jump are related to viruses, and we're going to talk about that. This happens in essentially all organisms. So this is what I started by saying, as is so often the case in science, you discover something peculiar in the organism you happen to work on, and you find out it's an enormously important general property, in this case of genes. Jumping genes can copy themselves and can accumulate in huge numbers, like 100,000. Okay? In jumping, they can wreak havoc, destroying other genes. Now, all of you know some kind of parasite. And you know the principle that good parasites do not destroy their hosts. Because if they do, they will go extinct. So a smart parasite is in some sort of balance with their host. They will take it over in some way, interfere, make the host serve them in some way, but they won't kill it. So if too much of this jumping happens and the DNA is destroyed, then they're killing off the host cell. So there are ways in which these jumping things are controlled so they don't overdo it and ruin the chromosomes. Okay, so too much of that an organism won't survive. Human genetic diseases can be caused by jumping genes, as you'd expect. If they jump into a gene and trash it, that's not a good thing. And in bacteria, the movement of genes that make the bacteria resistant to antibiotics can be promoted by their association with the jumping gene. They piggyback on the jumping genes and move around. And finally, highlighted in green, evolution is deeply affected by genome churning and dynamics, moving things around, and particularly by these mobile genes. Yes?
So you spoke about frequency in the second point, in terms of frequency, in terms of jumping. What do you mean by frequency?
How often
Okay, so,
one of the genes that has the potential to do this will actually do it. Okay. All right, so let's take any other questions while we're, yes?
Any known cause of the reason they jump?
Okay, the question is, why do they jump? And it's partly a matter of survival. If they jump to somewhere else, and they may have, and they're copying themselves especially, they may have a better chance of surviving. So it's a sort of existential thing. They can be transmitted, as I'll show you in a moment, kind of like a virus is. So again, that's a survival tactic for these pieces of DNA, these jumping genes. Yes.
Does the jumping happen in only one cell or in all simultaneously or something?
Okay, great question. See, you are really smart. Okay, so does it happen in one cell at a time, or is there some sort of coordinated jumping? It's generally a fairly random process that'll happen in one cell, and I'll show you this in a moment, and not in all of them at once. But there are certain things that can trigger more massive jumping. Think of it like musical chairs. The music starts and there's lots of stuff and the music stops. What is the music? For example, X-rays will do this. Things that cause genetic damage will trigger more rampant jumping. It varies a lot with the particular kind of jumping gene. Yes?
Does the whole gene jump, or can portions of it jump, causing these repeats?
Great question. Does the whole gene jump, or can part of it jump? Part of it can jump. And it can take neighboring genes with it. It gets pretty vicious. Yes.
So the system is expanding, or is it also?
The question is the system expanding, meaning are you always getting more copies? The tendency is to get more copies. There are rarely any purges, because there's no real mechanism for that, which means over time, all of us who have these things accumulate them. And we'll talk about that more in a second. Yes?
Pretty much all 20,000 genes jump a little bit?
No, not in any regular fashion. There are particular genes, as I'll tell you more now, that have the ability to jump, and I'll show you why. Yeah?
So the prerequisite for jumping is the cell division, right? You have to have cell division to jump, or can it happen instantly?
It can happen without cell division, but for it to be sort of propagated through an organism in some way, you would want the cell where the jumping happened to expand into a clone of cells. A whole bunch of daughter cells form from that cell, and you'd see the effect in all of those cells. And we'll talk about that in a moment too. So these questions, let me do one more slide. I love it that you're asking so many questions, but let me tell you what a transposon has, the two key features of a jumping gene, because this is really about your question, do all genes jump? Okay? So here's a representation of a blue piece of chromosome, the DNA, and a section of that DNA is one of these jumping genes, and it's in green, which means go. And the arrowheads represent very specific, special ends of the jumping gene that are acted upon by machinery that cuts there and moves it. Okay? That's one thing you need, special ends. Okay, so those are the special ends that can be cut and rejoined somewhere else. The second thing you need is the machinery to do the cutting and the pasting, to pluck it out of one place, or the copying, because some of them, as you know, are copied. Okay? And those, there are variations on that machinery, but one of the remarkable things is these genes often carry the machinery within them. The transposon codes for the machinery for jumping. It takes along its own toolkit. And this is, the machinery is called transposase. There are other names. Transposase is a protein that snips at these green ends like the scissors show, so it must be true, and lifts the thing out or copies it, depending on the variation, and sticks it in somewhere else and joins it all up over there. Okay? It's a complicated chemical process involving a fancy protein. But that's what happens. And you don't worry about, for today, about the process. It happens. Now, okay, I want to go back. Let's see. Yeah, so the transposon codes for its own kind of springboard. All right, now there were questions, so let me, yes.
Do you ever find them to jump back to their original place?
Do they jump back to their original place? Sometimes, with certain kinds of transposons, they leave behind a copy of the ends, or sometimes even the middle, and there will be a tendency to jump back to where that residue is, because of the aspects of the molecular biology that I'm certainly not going to go into. Yes?
Can the genes jump from one species to another?
Oh, great question. Can they jump from one species to another? Sometimes. But often there's species specificity, and that's another thing. The entire recombinant DNA industry, huge biotech industry that grew up based on being able to cut and paste genes, came from looking at the way bacteria defend themselves against DNA that's floating around, including transposons. Because if you're a smart cell and there's some of this stuff swimming around you, you don't want it to get in. Or if it does get in, you want to get rid of it as fast as possible, because what's it going to do? It's going to jump into your genes and make a mess of things. So bacteria have elaborate defense systems to keep out foreign DNA, especially transposons. And we have special defense systems to keep out viral DNA, okay? It's part of our immune response. So these are things where it's a battle. It's a battle for the integrity of the genome, all right? And there are lots of ways that the battle is waged, preventing and allowing insertions. Okay, yes?
Can a gene be made up of multiple transposons? Because you said bits of them could.
Yeah. There can be multiple transposons kind of superimposed in complicated ways. That gets very messy, but yes, there was a question there.
Is there any reason to think that the propensity for gene jumping is increasing over time?
Is the propensity for gene jumping increasing over time? I don't know of any evidence for that, but there are certainly environmental influences in particular organisms, particular situations where there will be more or less jumping stimulated. So it's certainly possible. You were first.
Do the two ends come together when it gets cut out?
Sometimes. There are lots of variations on this. There are lots of different families of transposons. Some of them end up being circular for part of their lifetime, so then yes, and others don't.
Well, if it doesn't close up, is it like an empty shell there waiting for it?
Well, it stays linear. Okay, now, yeah, this is starting to get, I think, fairly specialized, so let's talk about it afterwards. Yeah.
Is there any tendency for the jumping gene to attach at the end of the chain that it came out of, or some other chain?
Is there a tendency to target the insertion site to the end of one chromosome or something like that? Generally, no. Generally, they're able to insert in a lot of different places. There are some exceptions that have preferences for where they will land. Yes?
Are there opportunities for this to be a good thing in evolution?
Yes. It is a good thing in evolution because it's essentially an eternal experiment in what might be better. And most of what happens is worse. But sometimes what happens is better, like duplicating a gene that benefits you in some way, and then that organism that has the duplicate will have a reproductive advantage and will be better, at least for a while. Okay, now we're going to up the ante a little bit on this transposon business. I said this thing can jump. Why can it jump? It has the right ends, and it's got the machinery encoded in the middle of it. This element can act on its own, doesn't need any help from anybody, and it'll jump. Okay, it makes a protein, the transposase, which turns around and chops out the ends and so on. Okay, this thing does not have a working transposase gene. It may have some other stuff. It has the ends, but there's no machinery encoded in there, and it's stuck. This thing has lost its ends. These are very sad. And it has this gene, but the gene has nowhere to act on. The protein encoded by that gene has nowhere to go. All right. That's when they're all in different cells. But let's take this situation. Let's say we're going to put the two defective kinds into the same cell. Okay, now they're in the same cell. What do we have here? This gene becomes active and makes transposase, which is unable to act on these deficient ends. But it's floating around, and it goes, aha! And it chops out that one and moves the other one. So machinery coded for by one of the copies can act on the ends of the other. Okay, we're restoring the music. The music's coming from here. People running around, not in the chairs or up there. You don't want to stretch that one too far. Okay. Okay, so that one can jump. All right. So let's talk about musical chairs. As I mentioned, the transposons can be numerous. They can be tens of thousands of copies. Some of those copies have bad ends. Some of those copies have bad transposase genes. But there can be a whole bunch of good ones. And then there can be ones that have only the ends and nothing else. And in a cell like this, where there is no transposase gene anywhere, everybody stays put. And no genetic damage is done. It's very quiet. There is no music. Okay? Now, if instead there is even one copy that is complete and has the gene for the machinery, for jumping, then you can jump. Okay? And in fact, all of them can jump. Okay? And they'll do it at some rate, and they'll make an absolute mess of things. Okay. All right. So, look at that. We're making progress. Thank goodness. Okay. So, the point is that if in some manner a quiescent situation, like no transposase around, changes, for example, by mating two organisms together and one has it, one doesn't, suddenly things go crazy and you get all sorts of new mutations. And Barbara Mcclintock observed that kind of thing in corn. When she combined certain strains together, both of them were perfectly reasonable and well-behaved on their own, but when mated to each other, something went crazy. And we'll talk about Barbara's work next, talking about plant transposable elements, where this all came from, okay? Starting in the 1940s, building on work that goes all the way back to 1914, in fact, with some early observations. And Nina Fedorov, who was a Carnegie scientist, staff member, says it is perhaps an understatement to say that Mcclintock's early communications describing transposition were not widely hailed for their explanatory power. Indeed, she commented in the introduction to her collected papers that the response to her first effort in 1950 to communicate her discovery of transposition in a journal with wide readership, the National Academy of Sciences Proceedings, convinced her that, quote, the presented thesis and evidence for it could not be accepted by the majority of geneticists or by other biologists. It was very controversial, even though she was already a very famous geneticist. It just didn't, this is the Lincoln Memorial is in between the Washington Monument and the Capitol building. It's really disturbing, but she had the last laugh. Okay, so she got her PhD at Cornell in 1927. She was famous for her studies of maize corn. She showed the relation between chromosomes recombining and the phenomena you see in the microscope, where you see them in sort of X forms. She showed the importance of the middles of chromosomes and the ends of chromosomes. She was the third woman elected to the National Academy of Sciences in history in 1944. She was 42 years old. And the same year, she was elected president of the Genetic Society of America. And I mention this, this was years before she really went public with, there are jumping genes. And so she was a very well-established, highly respected geneticist at that time. But she was so irritated by the response to her, you know, people are saying, this is nuts, right? So in 1953, she stopped publishing about these jumping genes. She said, sort of, the hell with them all, and got the Nobel Prize for it in 1983. So kind of nice to finally get what she deserved, but really kind of sad that it took 30 years. So what did she find in corn? She found a gene or a place on a chromosome, turns out to be a transposon, called, she named it dissociator. And in the situation in corn like this, where there's a copy of dissociator, nothing happens. But in a corn cell like this, that has dissociator and another gene called activator, which you can imagine what that is because you just saw it with the music, okay? When activator is present, in this case what happens is not only jumping, but it actually causes a break in the chromosome, which is one of the things transposons can do. It's a nasty trick. And so you end up with this, where due to the presence of activator, there's been a break here. And the transposon may have jumped somewhere else. She had no way of telling at that time, okay? So that was an amazing phenomenon. This is the sort of machinery provided here, we now know, and this was the ends provided here, and it caused chromosome breaks. And she started studying that, and then found that dissociator could actually be in different places. So it's like a weak point that created almost in chromosomes that causes breakage. Okay? But that's not the only thing it causes. And the other thing it causes is patterns like you see on these individual corn plants, which each kernel is a plant. So you see these gorgeous patterns, much celebrated just for their beauty, and they have something to do with this too. The molecular biology attack on this problem was led by Nina Fedorov, who was a Carnegie scientist. So we've got this great tradition of outstanding scientists who worked on this at Carnegie. So she isolated these things. Okay, so what's going on when you have these little sectors? You have some solid colors and you have some patterns. And these differences are due to jumping genes. All right? And we're not going to get into all the details here, but we're going to say that there's a gene that's needed to make a protein that makes purple. Okay, so when that gene is working, you get a kernel like this. Okay? There it is. See, it's got an arrow. So it absolutely is true. And here, we have a different situation where there's a transposon stuck into this gene, destroying its ability to make the protein that makes purple. And with something like that, where it's really not working at all, you get this interruption and you get something like this. Okay? If it happens early in the growth of this thing. If it happens later, then some cells will have a working gene and will make the maroon and others won't, and you get this kind of partial pattern. Okay. So let's look a little bit more at that just to try to make that more confusing. We have a gene here that's working in some cell to make purple. Everybody's fine. And that cell will grow up to make lots of cells, and they're all still purple and everybody's fine. That's the simplest situation. Now we're going to make it more complicated. We're going to have a cell that has the gene in it and there's a transposon in it, so it can't make purple anymore. All right, so that divides, and you get a whole bunch of cells from that, and then another one, and they're dividing and growing. And all of a sudden, we see a purple cell, which is either a mistake I made in preparing my slides or I'm trying to tell you something. And what I'm trying to tell you is that in this cell, the transposon left. It jumped out. I don't know why it jumped out. It could have been a little radiation. It could have been it was bored. We don't know, but it does at some frequency jump out. Now all the cells, so it's, okay, so we go, it goes away, and now we have an intact gene again, and that's why we have a purple cell. So then all the cells descended from that one will be purple. And here it happened again. another one jumped out. And someone asked earlier, does it happen in all the cells at the same time or not? No, it doesn't. It's kind of a random process. And so now all the cells descended from that one will be purple, and you get purple stripes. Look at that, they're purple stripes. And in fact, right here, right there, is that first cell. Okay? So these patterns, which plant people call variegation, and they look like this, and they look like this, are clones of cells that have descended from an original cell where a gene jumped. Okay? This happens in you too, it's just not so visible. But if you spend enough time in a pot, you will reveal it. Okay, so I know you've been wondering about this. And the fact is that these observations, which were the first observations of this kind of phenomenon, well, the phenomenon had been seen, but it hadn't been understood. What Barbara Mcclintock did was figure out what was going on. And she figured it out before we knew anything about the structure of DNA. Just linear chromosomes and genetic maps, and something funny, right? So we as researchers in developmental biology work with fruit flies a lot, a classic organism to study chromosomes and development and so on. And as in corn, there are lots of transposons, and they behave in the ways I've been telling you about. So we'll forego the music this time, but you get the idea that if you have a bunch of transposons and you add something that has a transposase gene in it to what was a static situation, now things will start jumping and you'll get new mutants. In flies, it turned out you could do this by crossing certain strains of flies with other strains of flies. Some of the strains of flies, like this little guy here, have active transposons in them, good transposons with the red thing and everything. But there is a system for keeping things quiet in those flies, so nothing much happened. And then you have other flies that don't have any active transposons in them, so of course nothing's happening in them either. But if you introduce an active transposon from this fly into that fly, or into the progeny of both, now things go nuts in just the musical chairs kind of way. So this is how you combine things if you're flies, and not just if you're flies. And what comes out of that is a whole bunch of weird looking flies. Because in that situation, the transposase found it had lots of things that could help jump, and they started jumping all over the place and making a mess of lots of genes. Okay? So that's flies. And now we're going to talk about antibiotic resistance. Now, this is an enormous health issue right now, and the sweeping through populations of bacteria of resistance to all of our useful antibiotics is an extremely frightening thing, to me anyway. And more you find strains of bacteria that infect us, or animals, or even plants, that are resistant to most of our arsenal of antibiotics. The resistance depends on genes that code for proteins, and the proteins often, in one way or another, destroy the antibiotic, or they're the target of the antibiotic, but they change so that they're resistant to it. So they're genes. Okay, so we're going to start simply with this. So a bacterial transposon also has a transposase gene. I should have made it red here, but I got lazy. And then in the same stretch of DNA between the endpoints will be some of the genes for antibiotic resistance. There it is. Okay? These are bad. Now, because this thing can jump, and it can also, someone asked the question earlier, get to new cells, and I'll talk about that more in a moment, this thing can move around, and along the way it can pick up other resistance genes until you have a whole string of them, each one for a different antibiotic. It's all part of the mobility of the genome. So transposons can carry one or several antibiotic resistance genes. And once it's like that, that DNA gets loose on the world and is sucked up by bacteria during an infection or something in us, and this is really bad news. Okay? So Barbara Mcclintock's discovery in corn really revealed how some of this movement of resistance is happening. And while we generally credit sharing of things where basically new information is appearing in different organisms as it's transmitted in this or other ways, in this case it's really bad. Now in a population of bacteria that carry, and I mentioned the DNA is sometimes circular, one of these pieces of DNA with an antibiotic resistance gene, if you add the drug, the antibiotic that here is diagrammed as red resistance, then all the bacteria will die except the resistant ones, and the resistant ones will grow up and make you miserable. Okay, that's what happens. And the more there is sharing, okay, which can happen between cells, and even within cells making duplicates to make them even more resistant, pretty soon you have cells that are doubly resistant, and then sometimes the circles can join to form one circle. It's really a problem. Okay, so this is what happens. Third, consequences. We're whipping through the consequences here. Third thing is us. So here we have a paper published by this person right here, Maxine Singer, who was one of the pioneers in investigating mammalian transposons. Maxine, as I think all of you probably, many of you at least, know, was president of Carnegie. And so yet another part of this wonderful tradition of pioneering work to do with transposons at Carnegie. So these particular ones she was studying are enormously abundant. One of the line transposons, long interspersed sequences, there's 100,000 of them in one of your cells. That's amazing, right? That's a parasite, and all of you have it. Don't squirm. Okay. But good news is they don't jump much. They're boring. Okay? And it's a good thing they don't jump much, because you can imagine what would happen to the other genes if they did. However, they do jump some. They are one of a class. These are diagrams of different variations on transposons that are found in humans in various numbers. And that particular one, that line one, at some point in certain people, a person jumped into a gene that is necessary for your blood to clot properly. So the transposon landed in that gene, and molecular biologists isolated the gene from the people who had this problem, and these people had hemophilia. They can't do blood clotting properly, from a new insertion of one of these line sequences into this particular blood clotting gene. Okay, so it splits the gene, makes a mess of it, you can't do blood clotting. All right? There are lots of different transposons in us. They don't jump that much, but when they do, they can cause problems. And here is the really astounding part. Half of your genomes are made up of transposons. Half. We thought we had this special genome, making the cortex. We can think. Why do we have all this stuff in here? We're carrying around. Somebody asked earlier over here, do they get purged somehow? They don't. They accumulate. They don't do much harm. A lot of them sort of go to sleep. They mutate. They can't jump anymore. So they accumulate. It's a bunch of junk. It's not all junk. Some of them have some functions. But they've accumulated to such an extent that we're walking around with half our DNA being parasite DNA, with our DNA carrying in our chromosomes genes that jumped in your ancestors. You've got records of your ancestors' infections in you. You can study human history by looking at old transposons, if you wish. Or you can read about the Civil War. Okay, so that leads me to this. Look at that, we're almost there. Sorry, I'm taking a little too long here, but transposons and viruses, all right? Transposon DNA, they have a structure, and the proteins they encode for jumping have a structure, a sequence of amino acids. You can look around and say, does anything else look like either of those things, like the DNA sequence or like the proteins? And the answer is yes. And when molecular biologists got their hands on the transposons, and other molecular biologists, virologists like Maxine, got their hands on interesting viruses, lo and behold, there were all these similarities. So, the DNA of transposons is similar to something that's very familiar to biologists and family doctors, and we say, if it looks like a duck and jumps like a duck, maybe it's a virus. Now, what is a virus? A virus is a parasite too. You can think of it like a transposon, meaning chunk of DNA, that's in a little package that does quite well for going between cells, as you know, much to your dismay. So this happens to be a picture of the HIV virus, particularly nasty one. Now, it doesn't actually have DNA, it actually has that related molecule RNA, but those are nitpicky details. The main point is that in the sequence of that stuff is similarity to transposons, and a lot of the machinery that goes on inside the cell, including inserting the DNA into chromosomes, is very similar in viruses and in transposons. The transposons can act on their own as pieces of DNA, or they can be packaged in a viral coat, and which protects them, and then they're more infectious at a distance. They can infect other cells, yes?
Is that similar or connected in any way into the piggyback transposon used for similar genetic, like transfer between species?
Yes.
Or the baculovirus, which is being used in aerosol operations as a pesticide?
Each of these, you're just describing a variety of variations, different classes of transposons and viruses, some of which are used for engineering purposes. And indeed, two Carnegie scientists, Alan Spradling and Jerry Rubin, engineered the fly elements as tools for introducing genes into flies. And similarly, some of the ones you've described and others are engineered because this jumping ability can be very useful if you want to introduce a gene into something and test what it does, or change it in some way and test what it does. So as is true of all genetic engineering, it raises questions of, when you get beyond science, of what's ethical and what's a good thing to do and what's a bad thing to do. But these are definitely powerful tools for engineering various kinds of changes. So let's look at the life cycle of a virus to give you some idea. So here's a little virus. Here's a cell. It's going to stick to the outside of the cell, and inside the cell is a chromosome. There's an entry process that strips off the outer proteins from the virus. It goes inside, and now you have the virus DNA inside. That virus DNA can insert into a chromosome, and now it's there. Now it could jump out again, but if it stays there, or if it gets damaged and gets stuck there, it'll be inherited by all the cells that form when this cell divides. If, however, it's going to make more viruses, there's a copying machine that copies the viral DNA. There's in the viral DNA is the code for the virus parts, the protein parts, and those two things combine, and new viruses come out of the cell, sometimes breaking the cell open, sometimes going through the surface of the cell. And that's a viral life cycle. Okay, yes?
Are phage
bacteriophage, yeah.
Do they have anything to do with this?
They do. They are bacterial viruses. They come in many flavors, some of which have properties akin to this. Yes. Okay, now if we look at this process in people, we have an infection happening here, and that infection could, say, just be in your skin. If it's just in your skin and you have babies, you're not going to pass it on genetically unless you do something really weird. Because the only thing going to the babies is sperm or eggs, right? In terms of that kind of transmission. But if it gets into sperm or eggs, then it's passed on, that germline infection, the germline is sperm or eggs, and then it'll be inherited. And a person derived from such sperm or eggs, the whole body will have that viral DNA in them. Okay? And over here we have horizontal transmission, which is normal infection through a crowd. Please nobody sneeze. Okay? So both of those things can happen, and this is how, when it gets into the germline, that's how you start accumulating in your chromosomes the infections of your ancestors. If it doesn't get into your germline, then it's a clean slate and you get only new stuff. It's not inherited that way. Look at this. We're finally here. And what I've shown you is that growing out of Barbara Mcclintock's discoveries, and since then work of thousands and thousands of people to try to understand this, is that the static view of the genome, which by the way never completely made sense, and I'll come back to why, the view that the genome, the genes are just sitting there, never moving, and they're just copied and passed on generation after generation, that's wrong, and that there is the potential for genes to move around. The reason that makes sense is because when you look at organisms comparing, say, us and macaque monkeys, or us and turtles, I said earlier, you see that, a lot of things are similar, but there are quite a few changes. Well, that had to happen somehow, and these jumping genes help it happen because, as I mentioned, they can carry pieces with them, they can cause chromosome breaks, they can do all sorts of things to stimulate some movement of pieces of DNA and pieces of chromosome. Now, if that happens too much, it's a very bad thing. It will kill the cells it's happening in, and certainly will kill the organism if it's an essential process. You won't get a good egg from somebody because the genes are a mess. It won't work. Okay? So it's actually typically kept down, like any good parasite, it's kept down to a low level. There's small changes. Some of them are harmless. Some of them are harmful. Those are lost from the population. And others might confer some kind of benefit by moving genes around in some way, or even joining two genes together to create a sort of new hybrid gene. Okay, so that's point one about the evolutionary process, because that's what that is. The changes in genes are evolution. But now I want to talk about evolutionary trees and whether it's the right idea, whether a tree is really a web. Now this is what you're familiar with. There was some ancient organism that started changing and went through all these branches. It's nice and tidy. These are the main branches, the eubacteria, like the ones you're mostly familiar with, the archaea, things like that exist deep in oceans and other places, and animals and plants on the top sprout there. This is the view of things we've had, but if DNA is moving around, and somebody astutely asked the question earlier, does it go between species? And they might be closely related species or distantly, but if there's a transfer here, the tree is a web, not a tree. And you can get very rapid evolution where something can pick up, in essence, some information, an idea, something suitable from some other organism. Now, Strangler Fig, some of you have probably been in the jungle where there's amazing trees that engulf other trees, and over 100 years will grow up around them and completely destroy the tree inside. These are fig trees on the outside, and whenever their branches or roots meet up, they fuse. So that's kind of what we're talking about here. Here's my daughter standing beside one. That in this tree of life, if there's lateral transfer of DNA between different branches, you get a mixing up of things that make things a lot more confusing. Now, most of the time that doesn't happen. As I said, organisms have defense systems to keep out foreign DNA, but they don't always succeed, and then there can be picking up of other genetic information. Now, early in the primordial soup, as we all call it, life was little, microbes, 2 billion years of microbes before anything really fancier happened, and DNA and RNA and proteins were evolving to be better and better machines. Lots of practice and refinement during those times, and places you can go in a submarine now, like deep smoker vents like these, might give us hints about what some of that life was like when the Earth was still hot. Around these smoker vents, the hottest places do not have many microbes, but the places just a little farther away do, where it's roughly at what at the surface would be boiling temperatures, and there's incredibly rich microbial life at these places. And those things surely have transposons and jumping genes, and they exchange DNA and so on. So these things are quite possibly where a lot of the rapid evolution happened that involved sharing different DNA. You get complicated life down there too. These things, these yeti crabs actually eat microbes for lunch. So what we're talking about here, and there are lots of deep smoker vents now, what we're talking about here is horizontal gene transfer, where things can be exchanged, and there is evidence that this has happened in history. Now I want to sort of finish by showing you an analogy of lateral transfer from a game where Notre Dame beat Stanford. Now I'm a Stanford guy, so I think this represents a considerable sacrifice on my part, although this play, which was remarkable, was called back for a penalty. But it was a Notre Dame play, and I think it was pretty impressive.
There was another play at the end of the first half that, had it not been called back, would have gone down as one of the most memorable in college football history.
The long ball to the end zone. Notre Dame has people there, so does Stanford, and it's intercepted by Notre Dame. And the first half will end on the Notre Dame.
Handoff.
No, he laterals gets the ball to Zivikowski, and Zivikowski's telling he laterals. Turns the corner. Ball's working the far sideline. He's inside the 30. No, the lateral. Zivikowski a race to the end zone, and he goes in.
Okay, so that's lateral transfer. Okay, I'm going to skip this quote and go back to our summary here, and just sort of repeat for you where we've been together today. Most genes stay put in specific locations. Some genes don't. They jump. They're related to viruses. It happens in all organisms. They can copy themselves. They can accumulate in huge numbers. They can wreak havoc with other genes, destroying other genes. They actually can sometimes activate other genes by landing next to them and turning them on, which we didn't get into today. Too much of the jumping and wreaking havoc obviously will kill the organism, especially if it's in the germline. Human genetic diseases like the hemophilia I talked about can be caused by jumping genes. In bacteria, antibiotic resistance is sometimes spread by transposons that have resistance within them. Those are also used extensively in genetic engineering of bacteria. And evolution is deeply affected by these mobile elements that have allowed genomes to change much more rapidly than they would by any other process. And it started out in the cornfield, watering stuff, and looking at funny patterns on leaves, and looking at funny patterns on kernels, like those, and trying to understand what it meant. So thanks for your attention. You were fabulous.
Matt has stamina for more questions.
Maybe
We
we
can
can have
answer
the
quite a
lights
few
on.
already.
There are people sleeping, so don't turn the lights on too much.
Sorry, wake you up now.
Do we have any idea how many of these jumping genes that no longer jump form regulatory elements in this so-called junk DNA?
This is a very sophisticated question here, so let me enlarge upon it slightly. The question is, these jumping genes sometimes have control elements that turn on their own genes or can turn on genes next to them. So if a transposon like that jumps in beside a gene that's normally off in a certain situation, it may turn it on in the wrong place. And that definitely happens, and it's hard to put a number on how often it happens, but for example, in certain cases of cancer, genes that promote growth and should be regulated properly so when you don't want growth, they're off, have a transposon land beside them, and now they're on when they should be off, and you get cancer from that. So that's one. There are many mechanisms of cancer, but that's one. So it certainly happens.
I was thinking more of a normal genome. In other words,
Yeah.
through evolution.
Yes.
These
Okay.
things that stop jumping, and now they do something
Right.
positive.
The answer is absolutely yes, that happens. In other words, the transposon jumps in, turns on the adjacent gene. That's good for the organism. It stays put forever. It's stuck there, and it becomes essentially a
Well,
piece of the
numbers,
gene.
The only issue here is the numbers. You know, I hear 2% are for coding genes and 98% are regulatory elements and the so-called junk DNA. Those numbers don't seem right to me.
It's very difficult, I'd say, to distinguish DNA that matters from DNA that doesn't matter, and the best way to do it is to watch it over time, because anything that has a function will not change very fast, and things that have no function will change faster. And so doing comparative studies, say of chimps and humans, for example, will identify pieces of DNA, sometimes transposon derived, that have been exceptionally well preserved, and you suspect they're doing something useful. Yes?
Question back there?
Okay, yeah.
Way back.
Me?
Yep.
You had just mentioned cancer. I was surprised not to hear more about cancer in the lecture. Do transposons and jumping genes not have all that much to do with different cancers, or?
There are not a lot of cases where they do. There are many other mechanisms of cancer, and some of them do involve chromosomes being broken and rejoined. And so what happens is analogous to what we were just talking about, that a little DNA element that turns on an adjacent gene is normally way over there, but it's brought next to a growth promoting gene, and now it turns it on when it should be off. That happens in, for example, some leukemias. And so chromosome breaks and rearrangements can cause that kind of thing, and transposons can cause it, and then mutations within a gene can cause it as well. So there are a lot of different possibilities, and transposons are probably not a major part of that picture. Yeah.
Way back there.
Are transposons the major element behind some of the microRNA they're using for the CRISPR technologies?
No, as far as I know, they're not. Yeah.
Over here.
A different line of question. Any prospect of, I'm very poorly educated in this area, any prospect of finding somewhat open-minded person in the Trump White House you could give us quietly on the side, off premises?
Yeah, well, all of us who are scientists at Carnegie are concerned because a lot of our funding is dependent on federal sources, DOE, NASA, NSF, and NIH. And certainly the kind of cuts we heard about today would be totally devastating to us along with everybody else.
Is there one on the other side there, or here? Go ahead.
Okay. I just wanted to follow up and compliment bringing up what's research from the 1940s and what makes corn kernels have the patterns they have, and then how it gets us to today now, and funding of primary science. I thought that was a really interesting component of your lecture.
Thank you.
And then I also had a question. Oftentimes when you hear about conventional genetically modified foods, organisms, not the ones through CRISPR, one of the big critiques is that this doesn't happen in nature, and listening to your lecture, it seems like when you're talking about the web, it actually does. perhaps with less frequency, and I was wondering if you had any comments on that.
The classical work in genetics of agricultural plants and animals, which have changed immensely due to domestication and genetic engineering, usually that was done using either just finding variants that were useful or breeding things under conditions where you would irradiate them or treat them with other mutagens to try to induce a faster rate of mutation, and then looking for something that was useful, something that made more protein or something that had different fiber or different color or different taste. The difference now is that in some cases, and it's still fairly limited, you know exactly what you would like to do to change something. And you can go in a much more targeted fashion and change that thing, whether it be with CRISPR or with some other less recent technique. I personally think that many of the things that have been criticized are, in fact, harmless or even beneficial, but there's certainly some things where I would never want to see certain kinds of engineering happening. So what bothers me is the sort of wholesale rejection of tools that could be very useful. If you read about golden rice, you'll see an example of that, where vitamin deficiencies can be cured by it, and it's been lumped together with things that scare people, and I don't think there's any reason to be scared of it, quite the opposite. So it's a set of complicated issues, but it's certainly, what you said is absolutely right, that the sort of genetic testing of the waters, sort of endless variations and movings of pieces around and so on is happening all the time as a natural experiment of genomes. And the ones that are a little better tend to reproduce more and survive.
I just wanted to follow up on your comment about the funding threats. There is this March for Science, which is a worldwide effort to support the idea of science. I don't know if you personally have a position on that, or whether Carnegie Science is going to participate in that event.
Yeah. The question is about the April 22nd Earth Day March for Science, which will will be echoing from Washington to elsewhere. Many of our scientists are planning to participate in marches here and elsewhere to express their support for science, and I think many friends of science, including friends of Carnegie Science, will be participating as well. So it will be, I think a community reinforcing affirmation of the importance of science for our economy, our security, our understanding of our origins, and just the beauty of discovery and of life. And it's a very, heartfelt, I choke up thinking about it. So, but whatever happens on April 22nd will have to be sustained with other types of actions to really make a difference. And I hope that some of those are really productive. We'll see what happens. A lot will depend on the general public speaking up along with the scientists.
Take one last question here. You've had your hand up for a while.
Does the understanding of the mobility of genes, in the way you've been describing, has it affected the overall theory of evolution, realizing that there may be a lot of loose ends we don't see in the fossil record that could have gone in some pretty interesting ways?
Yes. This Mcclintock's discoveries, even though she wasn't, she was very interested in evolution in essence, but so the discoveries have certainly led to a complete change in our view of how genomes change over time and what processes might give rise to genes that are better for some reason or other. But there's still a lot we don't understand about what actually happened. It's a different kind of science than, say, physics, where you look for general principles that probably operated, throughout history or something. And biology is partly here's what happened to happen, and it happened to happen here. And as a result, there was this whole, the Galapagos rose out of the ocean and they were colonized, and you get all these new species from that. And that is a historical event that didn't have to happen. And so biology has a historical aspect that makes it a lot of fun, but also somewhat harder to study because you're trying to extrapolate to events you don't even know about, like like all the stuff he studies, the birth of continents and the movements of continents, and which volcano went off which and which ant got crushed. So it's it's really a very interesting business. But there's no question that now with genomics we can trace the histories of genomes and make inferences about the history of biology that were totally inaccessible to us before. It's a very exciting prospect, as some of you I'm sure have followed the studies of Neanderthal DNA. Most of you have. I'm 2.6%. It's the part right here. And I'm sure you followed some of that. There's fantastic work in that area, and so human history is being studied in a completely new way, and it's fascinating to see how the genomics is agreeing to such a huge extent with what linguists have deduced about the history of human languages. Those two things, both of them quite quantitative sciences, are showing a lot of parallel findings. It's really interesting.
Seems like a good place to stop. Before we say thank you to Matt again, let me remind you that the next neighborhood lecture is April 27th. It's going to be given by the DTM staff scientist Connell Alexander. The title's Rocks from Space. Be careful and a little bit afraid. So we hope to see you there. Let's thank Matt again.